sister chromatids anymore. We're going to call them recombinant sister chromatids. Because while they're almost identical, remember sister chromatids are identical, right? Are they identical? No, because you have a little bit of the DNA from that dad molecule, right? You did that homologous recombination. Okay, so your sister chromatids or recombinant sister chromatids remain attached. What gets separated in anaphase, and how that's unique from anaphase from mitosis, what happens in anaphase 1 is your tetrads separate. Yeah, so this is the inner one. So draw this. And this takes me a while to draw because I'm trying to keep consistent with the colors so you can follow all the way to the end of meiosis too. So when you're like drawing, just try to keep consistent, right? Because they're, right? Like that's why I'm doing it the way I'm doing it, right? So here, these in anaphase, you guys, someone tell me who is getting split apart and then pulled away from one another? What do we call these guys? The recombinant, well no, the recombinant sister chromatids don't get pulled away from one another because these are your recombinant, this is a sister chromatid pair, right? Sister chromatid. The reason we call them recombinant is because they're no longer exactly identical due to homologous recombination that happened in proteins. What gets pulled apart from one another is you can say homologous pairs get pulled apart, right? Homologous, these are homologous pairs, these are homologous pairs to each other, right? They get pulled apart, or tetrads. The tetrad gets, like, now no longer, it's no longer a tetrad, it gets pulled apart. Is that clear? Yeah? I mean, I don't think I have this for, what's it called, anaphase, like the polar microtubules elongate the cell, right? Because that's not like you already know that from prophase. From mitosis. You know that for anaphase with mitosis. But that is happening, right? But it's not like, so you know it's happening, but you wouldn't have to like tell me that's unique, because it's not unique to it. What is unique is, because if you look over here, where's anaphase? Oh, here's anaphase, right? Look at anaphase here of mitosis. Who is being split apart and away? What were these? Sister chromatids. Did we split apart these sister chromatids? No, they remain together. And so sister chromatids remain together. Why do we call them recombinant sister chromatids? Because they're no longer identical. Because of what happened? What made them not identical anymore? Homologous recombination. Is that clear? Okay. Now, so next we have, wait, do I wanna, actually, I think what I'm gonna do is, do that here, and then meiosis will be here. So telophase 1, let me make this cell a little bit longer now. What happens in, I guess just for purposes of things. In telophase 1, what is going to happen is that is somewhat similar to just regular telophase. Your chromosomes, these recombinant sister chromatids, right there, they do uncoil, right? It's not so important whether they separate from one another or not. They're going to actually reform just in the next phase, meiosis, right? But chromosomes uncoil. I'm just going to keep them kind of together. Into chromatin format. And then on this side. Chromosomes uncoil into chromatin format, nuclear envelope reappears, the meiotic spindle will break down. Right, and what else is happening? What is this process? Cytokinesis. Cytokinesis, right? And so by the end of cytokinesis, so this is end of telophase and end of cytokinesis, is you have two cells and you have here, maybe I will separate them. A little, now let me make this a little bit smaller like this. Okay. I should probably make this a little bit longer. Okay, so let's go through this. What is happening? First of all, how many cells do you have? 2 cells. You have 2 cells, right? If we look over here, let's look over here where we were here. You started with this immature sperm cell, 1 cell. How many DNA molecules? 1, 2, 3, 4, right? Which is the equivalent of what in human cells? Immature human sperm cells or egg cells. 46. You had a DNA molecule number 1 that sperm cell got from the father of that guy, right? And you have a DNA molecule number 1 that sperm cell got from the mother of that guy whose sperm cell this belongs to. Yes? So DNA molecule number 1 from mom, DNA molecule number 1 from dad, DNA molecule number 2 from mom, DNA molecule number 2 from dad, right? And 46 altogether, right? I mean 46. If we go here, now at the end of meiosis, we have 2 cells, 2 sperm cells. If we count the DNA molecule number, right? We have what? 1, 2, 3, 4. 1, oops, wait, I meant to draw. I'm making this confusing. Let me put the red here. That's this. Bless you. Bless you.
Thank you.
And then that's the blue guy, and then it's a little bit longer, just to distinguish them from the other ones. Okay, so what do we have here? We have 1, 2, 3, 4. 1, 2, 3, 4. So how many DNA molecules do we have in each cell? I mean, in the drawing we have 4. In an actual, it's still immature sperm cell, right? How many would this be? 4 is equivalent to what? 46, bless you. So this is 46 and 46. And remember how I said, however, this is called haploid already, and that's confusing, right? Because it's like, well, it's 46, so why is it haploid? Because diploid means having 2 representations of the 2 sets of chromosomes, right? Meaning you have a chromosome number 1 from mom, and I'm just saying chromosome, I don't feel like saying DNA molecule and chromatin formats, I'm just saying chromosome. Chromosome number 1 from mom, chromosome number 1 from dad, chromosome number 2 from mom, chromosome number 2 from dad, chromosome number 3 from mom, chromosome number 3 from mom. Now if I were to actually look, if you looked at this image, what do we have here? What is this long red one called? Long red one? What would you call that? The long red one. If red is mom. Right? And then this is chromosome number 1 from mom. Okay? And then do we also have a full-length representative of chromosome number 1 from dad in this cell? We don't, right? Do we have a full representative like we did here? Full meaning representative, the whole entire DNA molecule number 1 from dad in this cell? We don't. So that's no longer a diploid. Right? Because this one is mostly who? This is mostly mom. There's a little bit of dad on there due to the homologous recombination, but it's mostly the genetic information from that guy's mom. I mean, it's a sperm cell, right? That guy's sperm cell, right? But in terms of the DNA, right? Like it's mostly from mom, the genetic information. Is that clear? Yeah. And then if you look on this cell, we're talking about the DNA molecule number 1, the longer ones. Who do we have? What's this blue one? Who is that? Chromosome number 1 from dad. And this one, it's mostly chromosome number 1, also from dad with just a little bit of mom's information. Do we even have a full representative of chromosome number 1 from mom in this cell? No, so that's why it's haploid. You don't have the 2 full sets. Does that make sense? Of both from mom and dad. So I could do the same thing here, right? Chromosome number 2, this little guy right here, chromosome number 2, is from dad. This one is mostly also dad, right? This is the reverse here. Is that clear? Okay, so here we have 2 cells. What's gonna happen to these 2 cells? They're gonna then enter meiosis 2. They're gonna enter meiosis 2. Before they enter meiosis 2, there's something called interkinesis. Interkinesis, I don't need you to draw it, but just know that between meiosis 1, so all of this is meiosis 1, and what I'm gonna draw on the bottom here is going to be what? Meiosis 2. So this, all of this is meiosis 1. This is meiosis 2. That's going to be an M. Between meiosis 1 and meiosis 2, something called interkinesis happens. The only thing that you need to know that happens in interkinesis to these cells is these centrioles replicate, right? These centrioles, right, will make a copy of it themselves, right, here and here. So like if I were to draw this, nothing else happens here and here, it's just that the centrioles replicates. So then when this cell enters meiosis 2, and that cell enters meiosis 2, you have how many? Let me draw I'm gonna draw this cell on top and this cell on bottom. All right, so how many centrioles have here, maybe I will just also write interkinesis. So this arrow represents here and then down here, right? So interkinesis happens. Interkinesis, the only thing you need to know about interkinesis is that these centrioles copy themselves. So how many centrioles should I draw here? 4. 2, this is 1, 2 centrioles, so 4 together, because that's a centriole pair, another centriole pair. Remember, this is not one centriole. This, right, this is a centriole and another centriole, right? Yeah, so here, 4 together, 2 pairs. Yes? Okay. And then what am I going to draw here? I'm just randomly choosing this one on the left side. I'm going to draw this one here, and then the one on the right side I'm going to draw here. And then I'm going to go through meiosis with these. Okay? Any questions? So, let's see, blue and red. So I'm drawing prophase 2 right now. At the beginning of prophase 2, I'm going to kind of do it at the beginning of prophase 2. So this is prophase 2. Prophase 2 for both of these. We are in chromatin format. At the beginning of prophase 2, we're in chromatin format. And then what's going to happen in prophase 2? Same thing that happened in prophase 1, same thing that happens in prophase. What happens to the DNA molecules in chromatin? They condense into chromosome, okay? So, and then this is all that, and a little bit. Okay. So this is prophase 2, beginning of prophase 2. What happens? Meiotic spindle fibers form from the centrioles, meiotic spindle fibers forming, meiotic spindle fibers forming. So that's going to push away the centrioles to opposite poles of the cell. Opposite poles of the cell. I'm going to draw prophase 2 again, but now I'm going to draw. You guys, and for the interest of time, I'm just going to show you prophase 2, metaphase 2, anaphase 2, telophase 2 for this cell. But please imagine it also happening to this cell. And the same thing you can do for your drawings, right? When you do meiosis, of course, meiosis 1, you're drawing this, And then for meiosis 2, you can pick one of these cells and show me prophase 2, metaphase 2, anaphase 2, telophase 2. And at the very end, after cytokinesis, do show me what happened, but what's the result in here. But you don't have to keep drawing for this. Is that clear? Yeah, okay. So let me draw the meiotic spindle fibers on opposite poles. Let me do that. What happened to, can you guys remind me again, what happened to these chromatin molecules? They condense into chromosome format, and then these guys, these recombinant chromosomes now really, they do become attached once again to each other. So recombinant sister chromatids are attached at their centromeres, right? So I'm gonna draw, and I'm not gonna line it up in the center just yet. So here's this guy, chromosome format, and the sister chromatids are attached at their centromeres. Let me put this one here, chromosome format, and this is recombinant though. I'm saying recombinant, right? Because they're no longer exactly identical because of the homologous recombination that happened here, right? So the recombinant sister chromatids are attached at their centromeres. This is also happening here, you guys. I'm just not drawing it for interest in time, yeah? So this is happening here. Of course, the drawing here would be like different colors here, right? So recombinant sister chromatids are attached at their centromeres. The meiotic spindle fibers, those special ones that are called kinetochore microtubules, right? They attach now to the centromeres of each recombinant sister chromatids, and in a tug of war, they pull from one end to the other end, and once these end up in the center, we are now in metaphase 2, okay? Oh, what did I forget to talk about? Who disappears? So these meiotic spindle fibers can attach to the sister chromatids. The nuclear envelope. Yeah, so the nuclear envelope disappears. So metaphase 2, what am I drawing here? The recombinant sister chromatids are now in the what? In the cell equator. I'm not going to draw the polar microtubules, they're still there. The polar, the ones that are elongating the cell, they're still there. I'm just not drawing them. Okay, so who is now at the cell equator? The recombinant sister chromatid. If you look at meiosis 2 and compare it to meiosis 2, what's happening to meiosis 2 and what's happening to mitosis? It's almost like the descriptions. I mean, the beginning guys are different, the products are different ultimately, but the description of prophase 2, metaphase, anaphase 2, telophase 2 is identical to prophase, metaphase, anaphase, telophase of mitosis. Right? There's nothing, right? Your chromatin condenses to chromosomes. Sister chromatids attached to their centromeres. The meiotic spindle fibers attached to the centromeres of sister chromatids. Now, they're recombinant sister chromatids, we call them, right? But still the same, right? In metaphase 2, the recombinant sister chromatids are attached, not attached, well, they are still attached, but they're aligned at the cell equator. So then now that I've told you, right, that, what do you think happens in anaphase 2? What happens in anaphase 2?
They get pulled apart.
Who is they though? The recombinant sister, I don't know where, recombinant sister chromatids get pulled apart, which is what, anaphase of mitosis, what gets pulled apart? Sister chromatids. We're just calling them recombinant now because they're no longer exactly identical, right? So what gets pulled apart are your recombinant, we go this way, recombinant sister chromatids get pulled apart and then pulled away from one another. So they're going to get pulled this way, this way, this way, and this way. And please, remember, this is also happening here. And again, if you choose to draw this because it helps you, great, like for both beginning cells here, right? But you don't have to. Yes?
So is meiosis 2 basically the same as mitosis, except that the sister chromatids are,
Exactly, yes, and yes, exactly. Like this is what's happening, the descriptions are similar, except you call them recombinant sister chromatids, because sister chromatids, like in the term, it says it means like identical, because twin sisters, but they're recombinant because they're no longer they're no longer completely identical because of that homologous recombination. Exactly. Right. And so what's going to happen here in telophase 2 and then cytokinesis? So I'm gonna actually, you know what? I'm gonna draw telophase 2 and cytokinesis. Actually, no, I'll just do it separately, and then I'll separate them. So here are my chromosome number 1 that was from mom. It's going to uncoil into chromatin format. Chromosome, let me make it longer. Chromosome number 2, but this one is actually from who? From the dad, from the original dad of this guy, right? Uncoils into chromatin format, right? Chromosome number 2, right? Uncoils into chromatin format. Which one is this one mostly from? Also dad, right? And then chromosome number 1 uncoils into chromatin format, and this one is actually mostly mom. And then you've got, of course, nuclear envelope reappears. So let me reappear my nuclear envelope. Meiotic spindle disappears, right? And cytokinesis was happening. So eventually cytokinesis completes. So this is T2, telophase 2, and cytokinesis, the second cytokinetic event. End of cytokinesis. So you end up with what? I'm sorry, these cells look awful. You end up with these 2 cells here. And then, I'm not drawing this. I'm not drawing prophase 2 for this cell, but this cell, please, right? Prophase 2 is happening, metaphase 2 is happening, anaphase 2 is happening, and I am going to draw the final product. And I do want you to draw at the end, even if you don't draw meiosis 2 happening to both of these cells. I do at the very end want you, at the end of meiosis, I do want you to have 4 cells here. So then you can compare these 4 cells to each other, and ultimately the original parental cell, the original immature sperm cell. Okay? So let me draw, and I'm going to try to use the same colors over there. Clear envelope, centriole. All right, and then I'm just going to kind of separate this. Let's pretend the red one went here to the left. And this one is mostly red, a little bit of blue. And then let's go make this one. Go here, and then all blue here. Okay, so how many cells at the end of meiosis 2? Let me move this over here, because all we need is like that interface. How many cells at the end of meiosis 2 do we have? 1, 2, 3, 4. These are 1, 2 DNA molecules. What is that equivalent to? Right, this one, 1, 2, 3, 4, what is that equivalent to for our pictures? 46. So 4 is 46, 2 is 23, right? And then, so that's clear hopefully, right? And then, is this one identical to this one? No, right? You have DNA molecule, like this cell has a representative of DNA molecule number 1. Who is that representative from? From that, this is a sperm cell, right? Let's pretend these are 4 sperm cells, and that these 4 sperm cells are being made in his testes, right? And in one of his sperm cells, the DNA molecule number 1, the mature sperm cell, these are the mature sperm cell. The DNA molecule number 1 in this sperm cell, that's mostly genetic information from that guy's who? Mom. Is that clear? Does he also have does this sperm cell have the DNA molecule, any genetic information from, well, he has some dad's information, but off of the DNA molecule number 1. Is there any from dad? Do you see a big blue one? No, right? What about this? This is DNA molecule number 2. This one is all from dad. And then obviously I didn't do this with number 3, number 4, number 5, number 6, number 7. And why is it always going to be like, oh, like this is random, right? Like number 3, we don't know what number 3 is from. It could be just all from dad or all from mom, or a combination, like mostly dad, some mom, or mostly mom, some dad. Like how these get sorted is independent of each other and random. Is this clear? right? So this sperm cell right here, from that guy, he's making like billions of sperm cells a day, and this mature sperm cell, DNA molecule number 1 is from who? Mostly mom and a little bit of dad, right? And then DNA molecule number 2 is mostly dad. And see how this one's different from that guy? And both of these are different from that immature sperm cell initially, right? So I mean, I could keep going this, but hopefully, do you guys get this, right? And so this, like every sperm cell in the guy that's being produced, and every oocyte in the female that's being produced, is unique. That adds to the variability, right? Of the whole of our species, really. Like how you can also, you know how you have, unless you have identical, like twin sister, identical brother, right? How 2 different siblings can be so different from one another, even though they come from the 2 same parents. It's that due to that. Is that clear, you guys? So I know it's kind of tedious to draw this, right? But hopefully drawing it and following the colors.